Today we're talking about stem cells and division. Where it all began, you started as a cell smaller than a period at the end of the sentence. I know this looks very similar to the PowerPoint that we just went over that had to do with mitosis, but now we're going to talk about stem cells and development. And now look at you. So how did you get from there to here? And we're going to talk about that a little further. So getting from there to here, you're going from an egg to a baby. The original fertilized egg has to divide and divide and divide and divide. So there is cleavage. It is the repeated mitotic divisions of the zygote. So we know with mitosis, you're going from one cell to two daughter cells, and they have to undergo interphase, the G1, the S, and the G2 phase, and then they'll undergo mitosis, the PMAT, and then the cytokinesis. So the repeated division, you start up as a zygote, you develop into a morula, and then into a blastula. These are the first steps of multicellular development, and it establishes the future development of you as an egg into a multicellular organism. So to the right, we have a picture of a blastula. A blastula is a hollow ball of cells that is going to make up you in your mother's belly. So the blastula, like I said, is a hollow ball of cells. There's the trophoblast, which is the outside layer, and that's going to develop into the placenta and umbilical cord, which is going to attach to the mother. And then the stem cells are on the inside layer of the blastula underneath the trophoblast, and those are going to develop into the embryo. Stem cells are undifferentiated cells. Those are cells that have not been decided in what they're going to be able to do. Then we have gastrulation. Gastrulation is the folding of the blastula into tissue layers. So we have three tissue layers. We have the ectoderm, the mesoderm, and the endoderm. The ectoderm is the exterior and outer layers. This is going to develop into the skin, nails, teeth, and nerves. The mesoderm, meso meaning middle, is going to be the blood, bones, and muscle. It's also going to include your heart, the blood, and the skeletal and smooth muscle. And then the ectoderm, which is the inner layer, is going to be the inner linings of your digestive system and all of your different organs. So after fertilization, you start off as a zygote. The cell begins to divide and cleave, and you end up in the 8-cell stage. Continues to cleave, and you end up with the blastula, which is the hollow ball. This is what it looks like on the inside of the blastula. Gastrulation is the folding or the pushing in of one end of the blastula cells that will then differentiate into the different layers that are going to form the multicellular individual. So human development, these are just little pictures of the stages of development. So you start off as a little zygote. This is stage 1, day 1. Obviously, this is not to scale. You start off much smaller, kind of like the size of a period. It begins to replicate, multiply and divide. And then the zygote slowly grows from the zygote to the morula to the blastula to the gastrula, which is that 3 layers that are then going to develop into your insides and your outsides. So stem cells. Stem cells, like I said, are undifferentiated cells, so they do not know exactly what they're going to become. There are 3 types of stem cells. There's the totipotent cells, there's a pluripotent, and there's a multipotent. The totipotent cells are have the potential of becoming any kind of cell in the body. These cells are stem cells that develop early in are the early embryonic stem cells. So they are the initial cells that you start off with that are completely undifferentiated and are still figuring out what they are going to develop into. Then you have your pluripotent cells. These can become almost anything that is part of the body. The blastocyte, embryonic stem cells, and the fetal stem cells. Then you have your multipotent cells. They can differentiate into only a limited range of cells. These are the adult stem cells, and they're located in the blood, the muscle, the brain, the skin, and the tissue. So they're undifferentiated in the sense that they are stem cells, but they're located in areas like your skin or your blood. And when they do differentiate, they're going to differentiate into the specific cells of that area. So underneath your layer of skin, you will have adult stem cells that are undifferentiated, that when there is a need for them to grow, the stem cells, they will replace the skin that is sloughing off or missing. So the pluripotent stem cells, which is the embryonic stem cells or the fetal stem cells, start off in veno, so inside the body. This is what the embryo will look like, the 8-cell embryo. It'll form into the blastocyte and then will develop into the pluripotent cells. These pluripotent cells will then differentiate into the different cells of the body, whether they're the neural cells, the cardiac muscle, or the blood cells. So embryonic stem cells established from embryos shortly after fertilization. The embryo is separated into individual cells. The single cell from the embryo is placed in a dish and provided with nutrients and growth factors that stimulate it to divide. The resulting cell line will continue to divide as long as it is kept in a controlled environment and provided the appropriate factors to prevent differentiation. So this is a cultured stem cell, embryonic stem cell. So they fertilized an egg with a sperm, and they developed a embryo and let it divide and multiply in a petri dish in the hopes of using these stem cells to produce certain differentiated stem cells that the scientists would like to create. So adult stem cells are isolated from mature tissues. These are the multipotent stem cells, and they develop into the limited amount of cell types. So you have your hematopoietic, which are your different types of blood cells. So you get your white blood cells, red blood cells, your T cells, and your B cells. You have your mesenchymal, which give rise to your bone cells, cartilage cells, fat cells, and connective tissue. You have your neural cells, which give rise to your neurons, your astrocytes, and your oligodendrites. You have your epithelials, which give rise to cells of the digestive tract, and then your skin cells, stem cells, which will give rise to your epidermis and your follicles. So currently in stem cell research, doctors have been using stem cells to create trachea for cancer patients. So what they do is they use a trachea. They wash it, not necessarily with soap and water, but with different detergents to break down any of the connective tissue that belonged to the individual who initially had the trachea in their body. What they will then do is inject the stem cells from the individual who's going to receive the trachea. By doing this, they are making the cells in the trachea those of the body that it's going to be inserted in, so preventing the body from rejecting it and allowing the body to potentially recognize the tissue that is being inserted into the body. So this is your iPS cells. iPS cells are induced pluripotent stem cells. So we know that pluripotent cells are the fetal or embryonic stem cells. And then we know that multipotent stem cells are the adult stem cells. And adult stem cells are limited in what they can grow into. So what their scientists try to do is take those multipotent stem cells, reprogram them, so kind of rewind the tape where they were differentiated into certain stem cells where they were limited in what they could become, and make them induce pluripotent, so allow them to become undifferentiated again, to allow them to be differentiated into cells you would want them to become. So IPS stands for induced pluripotent stem cells, and they are differentiated stem cells that are created into undifferentiated stem cells to differentiate them into a specific kind of cell. So that is all we have in the stem cell realm. If you have any questions, please post them on Schoology, and we will discuss this in class tomorrow.